This is a really important MCAT topic. This is how a neuron activates a muscle cell. And I have a self-paced course that has over 500 pages of pictures like these and typed out explanations for all of ChemPhys and BioBiochem. If you're interested, the link is in my bio. Here we have the end of one of our neurons. And what it's doing is releasing a bunch of acetylcholine. The acetylcholine is binding to these blue proteins there. The blue proteins are ligand-gated channels. That means that when the ligand binds, and the ligand is acetylcholine, they're going to open up and they're going to allow sodium to pass through. As sodium passes through, that raises the voltage inside of the cell. Now what I want you to do is follow the membrane starting right here. This is the membrane of that muscle cell, and if you notice, it's going into the muscle cell. What that does is it creates a cave sort of structure. So if you were to fly a mini ship, you could fly into these caves without actually entering inside of the muscle cell because you haven't crossed the membrane to get inside the muscle cell yet. That means that all of the space inside those caves is actually extracellular fluid. And that's important because that means we have a higher concentration of sodium inside of these caves than we do inside of the cell, because you always have a higher concentration of sodium outside of the cell compared to inside the cell. That brings us to these red channels. The red channels are voltage-gated sodium channels. And if enough sodium is able to enter through the blue channels whenever acetylcholine opens the blue channels, that can raise the voltage enough that then the red channels open up and then sodium can travel through those red channels. Notice how these red channels line the entire cave. So if our first red channel here opens up, then that's going to raise the voltage in this area, which then will open up the second red channel. Sodium is then going to move through that channel, and that's going to raise the voltage in this area. And you can see how this is going to travel down the entire length of the cave. And the caves, by the way, those are called T-tubules. What the sodium does, and you don't have to understand this mechanism, but it opens up these purple channels that are on the structure inside of the muscle cell. These structures are called the sarcoplasmic reticulum, and they hold a bunch of calcium ions. When the sodium causes the purple channel to open, that's going to allow the calcium to leave the sarcoplasmic reticulum and be inside of the cytoplasm of the muscle cell. Once the calcium is in the cytoplasm, it's going to bind to something that are on these myofibrils. And let's zoom out and take a look at what that is. Calcium is going to bind to these orange structures, which are called troponin, and troponin holds the threads, which you can see that are like red and pink. Troponin is holding those threads onto this filament of actin, and those threads are called tropomyosin. Once calcium binds to the tropomyosin is going to shift where it's located, and it's going to reveal locations for myosin right here to then bind to the actin. So before calcium is present, myosin cannot bind to the actin because it's blocked by tropomyosin.